don't mean to interrupt your conversations or your lunch, but we have business to attend to, and we have time scheduled to worry about here. My name is Mike Castle, and I'm a congressman from the state of Delaware, and along with Representatives Bilbray, Capps, and Holt, we are the co-chairs of the Congressional Biomedical Research Caucus. This was founded in 1990, and it fosters interaction between important researchers, members of Congress, and staff. Let me thank all of you for being here. I think the subject matter that we're going to take up today, and the subject matter of this caucus in general, is vitally important to the future of healthcare in America, and it's important that people be listening. I'm not sure all members of Congress listen to all these things, so it's important that you're listening at this point. I would like to thank the Coalition for Life Sciences for hosting, and I'd like to thank Dr. Daley, who I'll introduce in a minute, for his perspective on stem cell research. Many years, not many, but several years ago, I had the occasion to visit him in Boston and see some of the work which he is doing directly. I can't stress enough the importance of NIH-funded research. When I talk to Dr. Daley about this, or other people involved in this, they always tell me about the NIH and the federal investment in research. It's critical for raising our standard of living in the United States and advancing our health. And frankly, the federal role is vitally significant in terms of pulling all the pieces together, and some of the sharing of information you may not get if you don't have the federal role involved in dealing with those issues. Many of us believe that in the area of either embryonic stem cells or some derivative thereof, we may be dealing with diabetes and Parkinson's and Alzheimer's and other diseases, hopefully sooner rather than later. That's really what this is all about. There are new scientific discoveries which we've all seen in the last few months or years, like the skin cell discovery, which you'll hear more about in just a moment. It just reaffirms the importance of all avenues of approach in terms of dealing with the whole stem cell issues that are out there. As far as I can ascertain, human embryonic stem cell research remains the gold standard. The legislation which Representative Vacanti and I have co-sponsored, which has been vetoed twice by this president. At one point, I had sponsored the only bill the president had vetoed. At another point, I sponsored 2/3 of the bills which he had vetoed. The same bill, they kept vetoing it. There is great belief that the next president will be more receptive to this, and that Senator McCain has voted for it, and Senator Obama and Senator Clinton have also voted for it as well, unless I'm surprised one of them is going to be the president of the United States here this time next year. There's an Energy and Commerce hearing tomorrow on this subject, which some of you may be interested in as well, and may want to at least watch, if not be able to participate in. Now, turning to Dr. Daley, he's the Associate Professor of Pediatrics in the Division of Hematology and Oncology at the Children's Hospital in Boston and Dana-Farber Cancer Institute. He is the Associate Professor of Biological Chemistry and Molecular Pharmacology at Harvard Medical School. Delighted, Doctor, I never had to take any of those courses. I gave her flunk biology myself. He received a bachelor's degree magna cum laude from Harvard University in 1982, a PhD in biology from MIT in 1989, and the MD degree summa cum laude from Harvard Medical School in 1991. He is board certified in internal medicine and hematology, and is currently a staff physician in hematology oncology at the Children's Hospital, the Dana-Farber Cancer Institute, and the Brigham and Women's Hospital in Boston. He has been elected to the American Society for Clinical Investigation, has received research awards from Harvard Medical School, the National Institutes of Health, the New England Cancer Society, the Burroughs Wellcome Fund, the Edward Mallinckrodt Jr. Foundation, and the Leukemia and Lymphoma Society of America. He received the inaugural NIH Director's Pioneer Award, a 5-year unrestricted grant to pursue highly innovative research. Dr. Daley's laboratory studies stem cell biology. He led a collaborative research team that successfully turned back the clock on human skin cells, causing them to revert to an embryonic stem cell-like state in which they can become any cell in the body. This is truly one of, if not the outstanding person in this country with respect to this whole area. And as I turn over the microphone to Dr. Daley, I must apologize because I have 2 other lunches at some point I'm going to have to run off to. But I hope all of you can say and hear all he has to say, and perhaps have some questions for him. I'll give you Dr. George Daley.
I want to thank Congressman Castle not only for that kind introduction, but for his tremendous leadership and support of the National Institutes of Health and his vision in this area of stem cell research, because I have had now about 20 years of experience as a medical scientist, and I can say that I've lived through a number of really, really exciting transformations. The discovery of the cancer-causing oncogenes, the sequencing of the human genome, and the establishment of human stem cells. And I have to say that I think the stem cell explosion is really one of the most exciting areas of biology today. It has enormous, enormous implications for medicine. As Congressman Castle said, I also take care of patients. I'm trained as an adult physician, an internist, a hematologist, an oncologist, but I'm at the Children's Hospital now. And I was drawn there because of the great questions in pediatric disease, especially the many inherited genetic conditions that lead to really devastating, chronic, debilitating diseases in kids. And stem cell research really is, I take a lot of my inspiration from the kids. And I just want to tell you a story of a kid that is emblematic of the kind of challenges we face in medicine. This is a kid that I saw in the emergency room about a month ago, and a kid with sickle cell anemia, a little 12-year-old boy. And as I walked into the emergency room, this little kid looked about 8 years old because the disease is chronic and debilitating, and often the kids don't grow as well as they should. This kid was literally writhing in pain, crying, and there were tears in his mom's eyes. And we had already hooked up the intravenous and been pouring the most powerful narcotic painkillers we have. And it wasn't for several days that I was able to get that kid's pain under control. I can tell you it is one of the most difficult diseases I care for as a doctor. And after years of seeing this kind of suffering, I still walk out of those kids' rooms and absolutely shudder. I can never become inured to that kind of pain and suffering. And yet I walk back across the street and I go to my laboratory and we're working on models. And the models are in the mouse and now in the human. Models that I hope one day will lead to a new type of treatment where we'd be able to take that child's own cells, change them back into stem cells, repair the gene defect, and then give that kid back healthy cells. That's where stem cell research is headed. Now, you may know that the Nobel Prize in Medicine was awarded to Martin Evans this past year for isolating mouse embryonic stem cells. And that was a paper he published in 1981. So we have had mouse embryonic stem cells for 27 years. And they have revolutionized research. So there's virtually no laboratory in modern biology that hasn't been touched in one way or another by strains of mice that are generated because we can make specific gene rearrangements in embryonic stem cells. But the field heated up 10 years ago when Jamie Thompson's lab isolated human embryonic stem cells. And human embryonic stem cells all of a sudden really announced to everyone that there was immediate human disease relevance to this research, and the fact that human embryonic stem cells derive from excess embryos that are byproducts of in vitro fertilization. This is what caused the kind of political and policy firestorm. And so what we're facing is a constant need in the scientific community to continue to educate about the value of embryonic stem cells in research. Now, what I'm going to spend most of my time today talking about is this new, exciting breakthrough, and we use that term rarely in science, but it's clearly a breakthrough, where we can take, and I can show you the biopsy of my forearm, and we can move these cells now into a petri dish and make them essentially the equivalent of embryonic stem cells. It's extremely exciting. But the topic is, do we still need embryonic stem cell research? And I am here to say absolutely, unequivocally, yes. So embryonic stem cells, which can grow in a petri dish, are probably most important as tools for research. Most of the debate has centered around their promise for providing cell-based therapies. And we had an entire symposium at NIH yesterday on the challenges and promise of cell-based therapies. And yes, most of us working in the field do believe that cells as medicines will become part of our armamentarium as physicians, but it may take decades to get there. Right now, these cells are tools for research. We can study human development and the genes that control human development by studying the way these pluripotent cells, these cells that are able to give rise to all tissues, form tissues in a petri dish. There is an increasing appreciation by the pharmaceutical industry that these cells are going to be important in drug development for testing toxicity. Think about it. We test a lot of drugs on animals, and that's imprecise. We can detect some toxicity in animals, but it's remarkable how many drugs end up being pulled off the market because once they get into lots of patients, they prove too toxic. Being able to test these drugs against human cells and human tissues is going to be highly valuable. But once again, much of the hype, much of the long-term promise is the creation of human tissues for transplantation. And so I'm often asked, how long is it going to take to get there? Because people want to know, when are cell transplants going to be curative? So I follow the literature like a good scientist. And so I'm perhaps not surprised to learn in one of the finest journals of medical research that already stem cells are transforming treatment. Well, I leave it to you to determine whether this is a successful application of stem cell research. She does look fabulous, that's true. But it's not just the National Enquirer. This is the New York Times Magazine. I know some of you may equate the New York Times and the National Enquirer. But what we have here is a legitimate advertisement for stem cell creams. These new and very expensive topical creams, emphasis on very expensive, they're not about putting embryonic stem cells in a jar and slathering them all over your face. It's really about exploiting the technology. Already, this sort of hype around the value of stem cells for rejuvenation, for creating, wrinkle-proof futures, is really claiming a lot of advertising space. I want to also point out that there's a risk to the hype around stem cells. And the risk is the creation of unrealistic expectations in the public. And that leaves desperate patients subject to exploitation. I get many, many emails from patients saying, what do you know about this clinic in Barbados? What do you know about this clinic in China? Should I fly to Portugal to have cells infused in my spine? There's a rising tide of stem cell tourism, which is really piggybacking on this kind of global interest in stem cells, and there's a risk behind this. So this year, I'm the president of the International Society for Stem Cell Research, and we have in place now a special task force which is coming up with a set of criteria, standards, guidelines, principles that we are going to ask the entire stem cell research community to follow in reasonably and prudently moving stem cells to the clinic. So the mission is to define those principles to combat this rising tide of stem cell tourism and to solidify the standards in the field and protect patients. So let me talk about cell transplants, because even though I do believe cells as models of disease are deeply, deeply important to medical research, much of the future promise is whether we'll be able to harness cells as therapies. And the chief roadblock to that application is the immune system. We realize from organ transplantation it's highly effective, but in many instances what we do is we substitute one disease, kidney failure or liver failure, for another disease, which is immune deficiency, because in order to get those organs to take in recipient patients, we have to poison their immune system. So a number of years ago, my colleague Rudolf Janisch and I reduced to kind of a mouse proof of principle, an experiment whereby we would create a customized, individual, autologous, self-rejection-proof stem cell using the methodology of nuclear transfer. Now, here was a mouse that has a genetic form of immune deficiency. This is like my patients, my kids who can't fight off infections. You take a little tiny bit of the tail tissue, you culture a skin cell, you take an egg, you move the DNA of the skin cell into the egg through this process of reprogramming, and that's an overarching term for this fascinating biological process whereby the specialized functions of the skin cell are erased and the pluripotency, or this tremendous versatility of the embryonic cells, are reawakened. And we create an embryonic stem cell in a petri dish that carries the DNA of the mouse. It is a self cell. Now, because the cells in the petri dish can be cultured and characterized, we can repair the gene defects and then create a healthy form of this cell, which can then be coaxed to become any tissue, for this experiment, the blood, and then transfused back into the mouse to treat the immune deficiency. This is a proof of principle. It really represents one of the long-term goals of stem cell research. The problem is that if we want to make this a platform for human therapy, we recognize that the key element of this reprogramming phenomenon, the egg, the human egg, is in very short supply. Human eggs are harvested for in vitro fertilization, and this is done tens of thousands of times a year, but their use in research is significantly limited. And this has driven the sort of a necessity among scientists to look for alternative ways to affect this reprogramming process. And indeed, many scientists studying embryonic stem cells and studying the biology that makes them unique. Why are embryonic stem cells kind of the tabula rasa of all cells? By understanding the sort of nature of embryonic stem cells, the goal has been to take the skin cells and directly transform them into embryonic stem cells. And that's what I'll talk about. As I said, there were many laboratories, including my own. In fact, my application to the NIH for the Pioneer Award was to do this experiment. But as is often the case in a competitive area of science, Shinya Yamanaka's group from Japan published first. What he did was he looked at the entire array of data that had been generated from many, many laboratories about embryonic stem cells and what made embryonic stem cells unique. And he said there might be a subset of genes, and he winnowed it down to just 24, which he believed would be essential to this phenomenon of pluripotency, this unique capacity of the embryonic cells. And he made a mini library of those genes, carried them into a mouse cell on a virus, and isolated cells that looked like embryonic stem cells. Now, when he characterized those further, he came up with a limited set of genes. These are genes that are master regulators of this embryonic potential. And also, interestingly, a couple genes that many of us might not have predicted, genes that are related to cancer. And as we now appreciate, there are tremendous links between the biology of this reprogramming and the de-differentiation that we see in cancer. And in fact, one of the sort of unanticipated benefits of this reprogramming research, I think, is going to be insights into cancer. So these cells, it turns out, if you inject them into an early mouse embryo, you create mice that have tiny little patches in all their tissues that come from these donor cells. Not full embryonic stem cells, but halfway. So a number of laboratories, Dr. Yamanaka's, Dr. Janisch, Dr. Hochedlinger in Boston, came up with improved methods of selection. And now in the mouse, we can actually generate these stem cells that will contribute to all the tissues in the mouse and even be passed in the sperm and can create whole mice. So literally, cells that are skin cells in the mouse can just a couple months later become an entire mouse itself. So we've been able to essentially make every skin cell have the complete potential to make a mouse. So think of the implications of what that means. So we're talking about the sort of age-old question about when life begins. Every cell, through this type of experimental manipulation, has the potential to make a life. So we have to ask real direct questions about whether we're talking about lives of a cell or lives of an organism. So the real question then were, would these factors be sufficient to induce the same effects in human cells? And my laboratory, as well as I would argue dozens, if not hundreds of laboratories around the world, were attempting these experiments. Initially, as is the case in medical research, there were many, many failures. In fact, we have failed to date on my first skin biopsy to actually make this work. But I have a second skin biopsy, and it does seem to be working. But what we did was we went back to embryonic stem cells. And we started from a human embryonic stem cell, one of the original lines that Jamie Thompson had generated. And we said, you know, if we make specialized, differentiated embryonic equivalent cells from these embryonic stem cells, these may be easier to reprogram. They may be just a little bit closer to the embryonic state. And so sure enough, these are cells in a petri dish that we get from the embryonic stem cells, but they look very much like the cells from my skin. And we introduce these reprogramming factors back into them in hopes of turning back the clock. And indeed, because our laboratory was very skilled and had a lot of experience deriving new human embryonic stem cells from discarded embryos, we were able to recognize the rare colonies in the petri dish that resemble the embryonic stem cells. So these are now coming from the equivalents of human skin. We can get them from fetal fibroblasts. We can get them from neonatal foreskin. It's a very good source of skin cells, and even cells from adult males. And these are just pictures that show that they share a lot of the markers, the physical characteristics of embryonic stem cells. Now, fascinating, we're learning a lot about these cells. It turns out that this process of reprogramming appears to decline with age. So that's a concern if we're hoping to make these cells, let's say, from elderly patients. And in those cases, there's still a lot of additional factors that need to be discovered. Are these cells equivalent to embryonic stem cells? They behave like embryonic stem cells in a petri dish. And we can do what's called a global microarray analysis. In other words, we can ask, what are the set of genes that these cells are expressing at any given time, and compare that to the set of genes that embryonic cells are expressing. And then we do this elaborate computer algorithm where we create a tree of relationships. And here is the input embryonic stem cell. And here are the reprogrammed, what we call induced pluripotent stem cells, or IP cells. And they all tend to cluster together, suggesting that they're closely related, but importantly, they're not identical. And they have the behavior of embryonic stem cells in that, like embryonic stem cells, they form a special kind of tumor under the skin of mice that allows us to see their potential to develop into lots of different tissue types, like certain types of retinal pigment cells, gut cells, muscle, cartilage, and the like. These are the gold standards we apply to human embryonic stem cells. And by these limited criteria, these human-induced pluripotent stem cells seem to behave similarly. Now, unlike many of the other claims of alternative forms of pluripotent stem cells, multipotential adult progenitor cells, which were put before Congress in 2002 as the reason to reject embryonic stem cells, fat stem cells, amniotic stem cells, all have made claims to pluripotency, but none have reached the same degree of scientific substantiation as these types of cells. And this is just to show that there are now 4 individual groups, Shinya Yamanaka from Japan, Jamie Thompson from Wisconsin, our group in Boston, and more recently a group from UCLA, which have been able to reproduce this work. So this is robust, it's reproducible, it's real. We just don't know. It's too early to know how powerful these cells will be. We now can go back to the mouse and redo that original experiment that Rudolf Janisch and I had done with the immune deficiency. Rudolf's lab has done the same with directly reprogrammed cells and now has a model of sickle cell anemia. And one of our goals is to be able to replicate this kind of success in the mouse with that kid that I told you about. My little patient who comes in with a painful crisis, we're looking to get stem cells and perhaps in the long term be able to do this type of cure. Now that is likely to take years, and I cannot guarantee you that these cells will ever be useful in treating a patient. I have been chastened enough by the failures in gene therapy, sort of the last great medical technology of the late 80s, which had the promise to cure dozens of genetic diseases. We're now 20 years later, and things are starting to work, but it may not be the panacea. So I am not here to promise you that embryonic stem cells or pluripotent stem cells are going to be the next panacea. But I can tell you that just as gene therapy has taught us an enormous amount about basic biology, the ability to take cells from any patient with disease and establish them as a tool in culture will transform medical research. And my group has already been able to take cells from patients with immune deficiency, Down syndrome, juvenile diabetes, 2 types of muscular dystrophy, Huntington's disease, Parkinson's disease, and a bunch of others. And we have pluripotent stem cells in a petri dish. That will transform our ability to study those diseases. But because, I remind you, we use viruses that can cause cancer, these cells are not useful for the clinic. And I cannot guarantee you that because this process actually activates the native genes in the cell, which can also be associated with cancer, that even if we learn how to do this process without these cancer-causing viruses, we won't establish a cell that might not have a predisposition to cancer. We just don't know. It's too early. So there are many, many efforts, and the next big breakthrough will be to achieve this without viruses. But a word of caution is that you may hear that there are certain types of research which, because of ethical concerns, research like nuclear transfer, that this should be done in mouse models and not done in human cells. But we have to be wary of extrapolating from mouse models. So in the last year, scientists at the National Institutes of Health, Ron Mackay, discovered that mouse embryonic stem cells are actually different from human embryonic stem cells. And so we have to have questions about iPS cells as well. We are just beginning to understand reprogramming. And in fact, I believe that the way that the egg reprograms a cell, which is faster than our genes, that this is a linchpin and a key aspect that will enhance the efficiency of making iPS cells. So I think understanding nuclear transfer remains an important goal. So embryonic stem cells, iPS cells, are they the same? Do we need iPS? Do we need ES? Well, I remind you that the knowledge of embryonic stem cells was essential to making iPS cells. The ES cells remain the gold standard. If we have questions that relate to the nature of stem cells themselves, the way they arise in the human embryo, we're not going to learn that by studying skin cells. The principles that govern the biology of the human embryo can only be studied with the human embryo. And these have enormous relevance to human disease, infertility, miscarriage, chromosomal abnormalities, and birth defects. I think it is a tremendous vacuum in our knowledge of human biology that we have such a poor understanding of the earliest days of human development. And that vacuum is because we have political opposition to funding this research through the federal government. And the consequence of that is the tolerance of infertility, miscarriage, prematurity, and birth defects. Stem cell biology, I'll finish with this. Stem cell biology is a new frontier. And it is by no means time to limit the range of options and the tools that are available to research as we push this new frontier forward. And I'll just leave you with an invitation to the meeting of the International Society for Stem Cell Research, which is being held in Philadelphia in early June. This is a convocation of researchers from all over the world where you will see a remarkable expanse of research in all areas of stem cells, embryonic, fetal, neonatal, and adult. And I think most of the scientists there would show broad support for pushing all these areas forward. And I thank you for your attention. I hope we do have some time for questions. Kevin? Yeah.
Dr. Daley, during the recent congressional debate on stem cell bills, there was a lot of talk in both the House and the Senate about adult stem cells in 72 to 75 diseases that have been cured. Why do we need to go any further if adult stem cells are as successful as these?
Yes, so when we talk about the success of adult stem cells in therapy, we're primarily talking about the success of bone marrow transplantation. That is, there are many, many dozens of genetic diseases of the bone marrow, like sickle cell anemia that I talked about, immune deficiencies, and then acquired conditions like leukemias, lymphomas, and the like. And bone marrow transplantation has been successful for some of those patients. But you have to look at the details. So just in the last month when I was on service, I saw several kids with diseases that we hoped we could treat with bone marrow transplantation. I had a young girl who was a vigorous athlete, and then she started finding that she didn't have the stamina on the soccer field. She came into the clinic. She was anemic, but she also had bone marrow failure. She had complete bone marrow failure. The first question I asked her, how many brothers and sisters do you have? Because I knew that my only chance of really curing her was a bone marrow transplant, ideally from a sibling. She had one brother. We tested the brother, he's not a match. Now instead of being able to do a curative transplant, we're looking for anyone in the world who might just by chance be a match to her, and her risk of the immunologic devastation of that transplant goes way up. If I could instead take a skin biopsy, make a stem cell, and give her back what would be her own blood cells, that would be the ultimate cure. So it's not that adult stem cell research isn't enormously valuable. It is, and it would be our first mode of therapy, would be to actually deliver adult stem cells. But they're not available to all patients. And for those, if any of you have had friends who've gone through a bone marrow transplant, you will know it's not a walk in the park. An autologous stem cell transplant, where we put back your own cells, is a lot easier. It's actually done as an outpatient in most settings. So we need alternatives to adult stem cells. Any other questions, please?
You mentioned the poor understanding of embryo development.
Yes.
And as that being part of the reason for the embryonic stem cell research. Can that be explored simply with stem cells, or do you have to engage in research on the whole embryo rather than just the stem cell?
I would argue that we need to be open to the prospect that the early human embryo that we are producing in vitro fertilization clinics to the tune of hundreds of thousands of years, hundreds of thousands per year, many of which are going to be discarded as medical waste. These are enormously valuable for understanding these early days of human development. And right now we don't do that. So I think I want to open people's mind to the range of questions that we're not asking, because there's a cost to not asking that.
I really appreciate your comments about concerns about overselling the technology and what's happening with the media lately. And I was really happy to see that you and all of your colleagues at the head of the field are now saying that it's could be decades before we see cures for this. But as an advocacy community, the battle has been waged between the ethical concerns and cures. This will provide cures. In light of the fact that it's really research for cures, what can be a change in strategy or a successful strategy, effective strategy in advocating for stem cell research?
Yeah, I think your question is so well articulated, is that in the haste of the sort of public debate and the spotlight, what often gets highlighted are, you know, the pictures of kids with disease, the wheelchairs, the notion that we're all really driving towards cures. And yes, in the long run, that's where we're all headed. I mean, essentially, this entire huge framework of medical research is aimed at understanding disease and really relieving suffering. But one of the challenges that we have at the NIH all the time is to educate people on the importance and value of fundamental inquiry. You just cannot know where the breakthroughs are going to come. You just can't. And I think a very good argument can be made that the United States enjoys such a remarkably successful biomedical research community, biotechnology industry, and healthcare industry because we've invested heavily in fundamental medical research through the NIH since the post-World War II era. It's a harder sell. It's harder to kind of get people to relate to, you know, understanding mitosis of cells in a petri dish or understanding how the stem cells form in an early embryo. It's harder to sort of see how important that is. It's easier to say, we're going to treat your grandfather's Alzheimer's. But we have to keep trying. And I think I certainly have made a major effort every time I've given a speech, and I think most of the scientists who've really spoken out a lot, to emphasize the value of fundamental inquiry, because that's not only going to help Alzheimer's, it may have unanticipated benefits in cancer and AIDS and the like. We'll keep making that fight.
Can you tell us a bit more detail about the functional differences you've seen between iPS cells and embryonically derived cells?
Yeah, so, you know, we're just beginning to do these side-by-side comparisons. And I would say it's far too early for us to make any real concrete claims. But we're testing hypotheses. So one hypothesis is that embryonic stem cells and iPS cells will be completely the same in all our assays of blood development. In fact, what we're finding is we have a mouse model where we've made stem cells by nuclear transfer, by a process called parthenogenesis, and by this iPS methodology. And we've compared the three. And in this limited set of experiments, by far the worst are the iPS cells. Now, we don't understand why. So that takes us to the next question. Well, maybe iPS cells from the skin have some memory of there being skin. Maybe they'll make skin better than blood. So our next test is to say, well, let's make iPS from blood and skin and then see if they have a memory of where they came from. We just don't know. It's too soon to tell. So obviously in the initial flurry of papers, we're arguing aggressively these cells are like embryonic stem cells. I mean, we want to say that because we want to get our papers published. And now we're sort of in the second wave going, okay, how similar are they? And we're asking the more detailed questions. We just don't know. I think, I hope, that we'll understand the reprogramming process well enough that we'll make them the equivalent of ES cells. But I can't tell you that we've gotten there yet.
You explained yesterday, I don't know if it was you, but someone mentioned that the iPS cells with tetraploid complementation were only able to make a mid-gestation.
I spoke about that. So it's a little bit of arcania, maybe, but I'll try to explain it as easily as I can. As I said, we have many more experimental tools in the mouse to give us confidence that iPS cells are very similar to ES cells. We don't have the same range of tools in the human because we can't do the same kinds of experiments. But even in the mouse, there's a fascinating experiment. It's one of my most, it's just one of the experiments that we do in our laboratory that just blows my mind. And that is, if we take a mouse embryo at the 2-cell stage and give it a little electrical shock, it becomes a single cell again, which has 4 sets of chromosomes. It's a tetraploid. That embryo cannot become a mouse. But if we, at the early embryo stage, now inject embryonic stem cells into that embryo, the embryonic stem cells we inject can become a mouse. So literally, we can start with a petri dish full of embryonic stem cells, let's say from a mouse with a black coat color, and we inject it into an embryo that's tetraploid, and we get, in a single gestation, we get an entire mouse. So literally, that proves that embryonic stem cells can become a mouse. When you do that same experiment with the iPS cells, you don't get a mouse. You get up to about mid-gestation, and then things fall apart, and we don't know why. Now, some might argue that we just need to get better at making iPS cells, and that ultimately we'll get to a mouse. And maybe that's true. When we get there, then we will be faced with the reality that every single one of your cells, if you will, if we want to make the leap to the human, every single one of your cells I could manipulate to make a person. So this whole idea about cloning, and we have to separate, when we're talking about cells in a petri dish and the value of cells in a petri dish for research, we have to separate that from this whole notion of how we respect life and all of that. Because with this methodology, every single skin cell, every single buccal mucosa cell that you spit into the sink when you brush your teeth has the same fundamental property. That is, with experimental manipulation, every one of those cells could be made into a complete being. It's not science fiction. We do it in mice.
2 years ago, Robert Montes' group described a method for isolating human embryonic stem cells without destruction of an embryo. I was wondering if you could comment on the uses and limitations of that method.
Yeah, I think it's an experiment that was done for political reasons and not scientific reasons. I don't think there's any scientific advantage to using that method. It simply shows that you can take a single cell and establish an embryonic stem cell from it. So I don't think anybody's using that as a method. It simply fueled the argument about the use of embryonic stem cells.
So the addition of oncogenic genes into a somatic cell limits the application of the iPS cell, potentially. But does anyone understand how the oocyte is able to reprogram a cell?
No. I mean, I think, on the one hand, the glass is half full, on the other, it's empty. There's been really decades of research aimed at understanding how eggs can reprogram, primarily in frogs, a little bit in mice, very little in humans. And there has been a lot learned. So we do know many of the principles. We know that when the somatic cell gets put into the egg, that there's this erasure of all of the expression patterns from the cell and a reawakening of all the genes that are relevant to the embryo. So there's preformed in the egg sort of enzyme complexes that are acting to remodel the input genome. Why is that? Well, because the sperm comes into the egg and gets remodeled. So it's fascinating biology. And, you know, I think this idea, so I'll tell a little story. One of the genes that Jamie Thompson's group showed reprograms is a gene called Lin28. We were studying that very gene in our laboratory, not for reprogramming. It was in a completely different project because we showed that it was relevant to this class of RNAs that are important in cancer. So this is Lin28. Turns out to be a key linchpin between cancer and the reprogramming of stem cells. And I think we're going to learn more and more that these processes share a lot. So it's, once again, another way that these unanticipated confluence of different research areas really move research that far forward. Another reason to invest broadly in basic research. How are we doing time-wise? Probably one more question, I guess. Yeah.
How much do we know about the effect of the telomeres and telomerase when we're doing IPS, and how is that affecting what we can expect from the IPS cells?
So it's a very, very interesting question. And in fact, one thing that one hypothesis about why this method is so inefficient. In other words, why did we fail on my skin fibroblasts and my younger postdocs' fibroblasts grew great and made iPS cells great? Well, one question is that, you know, I'm 47, he's 34, you know, I've had a rough life, and my cells had deteriorated and their telomeres had shortened, and in a sense, I was more senescent than his cells. And so one of the hypotheses for explaining why this is so inefficient is that you have to find cells that happen to have maintained the ends of the chromosomes. We just don't know. And I think it's a really, really interesting question. One of the potential limitations of iPS cells is that they, unlike the embryonic cells, which do seem to be completely rejuvenated for their telomeres and their mitochondria, the iPS cells may be a little sick. They may, you know, I mean, we're not as young as we used to be. And, you know, I can say that this observation that we've made, that it seems to be much easier to make iPS from the neonatal cells and the fetal cells rather than the adult cells, tells me that there's something about the senescence clock in cells that is relevant to this reprogramming process. Once again, lots to learn. I thank you very much for your attention. I'll be here to answer any individual questions you might have.